Public health authorities across Europe and Western Asia are reassessing the risk profile of the Puumala virus, following reports of a rare and highly aggressive mutation that deviates from the pathogen's historical behavior. For decades, the Puumala strain of hantavirus has been categorized by the medical community as the primary cause of nephropathia epidemica, a generally mild form of hemorrhagic fever with renal syndrome. While the virus has long been known to circulate among bank vole populations, its impact on human health was traditionally limited to acute kidney injury, fever, and blurred vision, with a case fatality rate typically falling below 1%. However, a significant shift in clinical presentation has been documented in recent medical surveillance reports, most notably in a fatal case study originating from the Moscow region. This specific variant, characterized by a complex genomic reassortment, appears to have gained the capacity to strike the respiratory and renal systems simultaneously. This dual organ targeting effectively bridges the gap between the two major hantavirus syndromes: the kidney-focused hemorrhagic fever, common in Eurasia, and the lung-focused cardiopulmonary syndrome, traditionally associated with New World strains. +2. The emergence of this mutation is particularly concerning for epidemiologists because it challenges the standard diagnostic protocols used in clinical settings. Normally, a patient presenting with Puumala virus would not be expected to show severe pulmonary distress. However, recent data indicates that the mutated strain can cause rapid progression to pneumonia, acute respiratory distress, and shock within hours of hospitalization. This specific development in the Moscow region, detailed in reports published as recently as March 2026, involved a patient who succumbed to the infection just 28 hours after admission. The clinical progression was described as unconventional, beginning with standard hyperthermia and severe weakness, but quickly escalating to include a heavy cough and shortness of breath, symptoms more commonly associated with the deadly Sin Nombre or Andes viruses found in the Americas. This development forces a reevaluation of how regional health systems monitor hantavirus outbreaks, as the mild reputation of Puumala may no longer hold true for these emerging sublineages. +1. The geography of the threat is also expanding. While the Volga Federal District in Russia remains a primary focus due to its high density of hantavirus-carrying rodents, the Central Federal District and surrounding European territories are seeing new molecular diversity. Experts note that the Puumala virus is subdivided into 8 distinct lineages, including the Central European, Finnish, and Russian variants. The recent identification of reassortant strains, where the virus exchanges genetic segments, suggests that the pathogen is evolving in nature during its constant circulation within host animal populations. For the public, the concern lies in the fact that these viruses are often transmitted through the inhalation of aerosolized dust, contaminated by the droppings, urine, or saliva of infected bank voles. As these rodents thrive in forest environments and rural dwellings, the intersection between human activity and mutated viral reservoirs is increasing, necessitating a more robust and transparent reporting system from international health agencies. The technical investigation into the Puumala mutation has centered on the virus's genomic structure, specifically the reassortment of the M segment. Genomic sequencing of the lethal Moscow strain revealed that while the virus remains endemic to the region, it has undergone a significant molecular shift. Researchers at the National Institute for Communicable Diseases and various European labs are analyzing how these exchanges between viral segments can lead to increased virulence. The data suggests that the exchange of the M segment, which encodes for the viral envelope glycoproteins, may be the key factor allowing the virus to better penetrate the vascular system of the lungs while maintaining its traditional affinity for kidney tissue. This multiple organ failure profile was previously a rarity for Puumala infections, but is now a documented risk. Laboratory reports indicate that the widespread damage to small vessels leads to increased vascular permeability, causing the fluid leakage that results in both pulmonary edema and acute kidney injury. +2. Parallel to the Puumala developments, global surveillance systems have been on high alert due to a separate but equally severe hantavirus cluster involving a different strain. As of May 2026, the World Health Organization and the European Center for Disease Prevention and Control have been tracking an outbreak of the Andes hantavirus aboard a cruise vessel, the MV Hondius, in the South Atlantic. While this outbreak is linked to a different species of hantavirus, it highlights the current intensity of global public health monitoring. In that instance, passengers presented with rapid pneumonia and shock, similar to the symptoms seen in the new Puumala mutation cases. The international response has involved authorities from the United Kingdom, the Netherlands, South Africa, and Switzerland, utilizing specialized biocontainment units for medical evacuations and intensive care management. The contrast between these events is critical for clinical accuracy. While the cruise ship outbreak is being managed as a localized cluster with suspected person-to-person transmission, a unique trait of the Andes virus, the Puumala mutation represents a shift in the environmental reservoir's baseline threat across the European continent. +2 data from the European Union's early warning and response system shows that human hantavirus infections typically vary between 1,500 and 5,000 cases annually in the region. However, these figures are likely underreported because the mildest cases of Puumala infection are often mistaken for the common flu. The danger of the new mutation is that it may bypass this mild phase entirely. Hospital records from the most recent fatal cases show that markers of viral replication and T-cell infiltration were found simultaneously in the kidney, spleen, and liver tissues during autopsy. The co-localization of these markers at the sites of tissue damage suggests an immunopathological mechanism where the body's own immune response, triggered by the mutated virus, contributes to the rapid destruction of organ function. This level of detail from recent pathology reports is driving a push for more comprehensive testing, including metagenomics and viral-specific PCR, for any patient presenting with unexplained respiratory and renal symptoms in endemic areas. In response to the evolving threat of mutated hantavirus strains, public health guidance is focusing on aggressive prevention and environmental surveillance. Because there is currently no widely available vaccine for Puumala virus that provides full protection against these new reassortant strains, the primary defense remains the limitation of exposure to rodent reservoirs. The bank vole, which serves as the natural host for Puumala, can reach infection prevalence rates of over 70% during peak outbreak seasons. Health officials advise that prevention is centered on cleaning and ventilation protocols in areas where rodents may have nested. It is critical to avoid sweeping or vacuuming dry droppings, as this action can aerosolize the virus, making it easy to inhale. Instead, the use of disinfectant sprays and wet mopping is recommended to keep dust particles grounded. For individuals working in agriculture, forestry, or those cleaning seasonal cabins, the use of high-grade masks and gloves is increasingly viewed as a necessary precaution rather than an optional one. +2 official statements from the ECDC emphasize that while the risk to the general European population remains low, the moderate risk for those in direct contact with rodent-infested environments cannot be ignored. Surveillance programs are being expanded to include more frequent testing of rodent populations to identify where new mutations might be emerging before they jump to humans. However, there are honest limits to our current knowledge. Scientists are still working to determine if the Puumala mutation that strikes both the lungs and kidneys is a stable, permanent change in the virus's evolution or a rare event triggered by specific environmental conditions. There is also uncertainty regarding the duration of immunity for those who have survived previous, milder versions of the virus, and whether that immunity offers any cross-protection against these more aggressive reassortant variants as the situation develops. The international medical community is calling for a unified diagnostic approach. This includes the use of an extensive respiratory pathogen panel combined with specific hantavirus serology for any case involving rapid-onset pneumonia. Public health agencies continue to monitor travel routes and shipping logistics to ensure that any potential clusters are identified and contained quickly. The goal is to prevent the kind of rapid, undiagnosed progression seen in recent fatal cases by ensuring that clinicians are aware that kidney viruses can now present with lung symptoms. For now, The focus remains on vigilant monitoring, transparent data sharing between labs, and public education on the realities of zoonotic disease in a changing environment. For more updates on global health surveillance and emerging viral threats, consider subscribing to this channel. In the laboratory environment, the focus has shifted to the molecular architecture of the Puumala variant, specifically following genomic sequencing results that have identified a critical reassortment in the virus's genetic makeup. Scientists at the National Reference Centers and Regional Virology Labs in Russia have confirmed that the fatal strain identified in the Moscow region is a reassortant, meaning it has undergone a genetic exchange between different viral lineages. Specifically, the research points to an M-segment exchange, which is the part of the hantavirus genome responsible for encoding viral envelope glycoproteins. These proteins are the primary tools the virus uses to enter human cells. By swapping this segment, the virus appears to have altered its tissue tropism, moving beyond its traditional target in the kidneys to aggressively infect the pulmonary system. Laboratory findings indicate that while the viral segments are endemic to the Central Federal District, this particular combination has created a clinical profile that mimics the far more lethal hantavirus pulmonary syndrome usually found only in the Americas. The clinical progression recorded in recent hospital data underscores the diagnostic challenges posed by this mutation. In the landmark case study from the Moscow region, the patient presented with symptoms that initially mirrored a severe flu or standard Puumala infection, including hyperthermia and extreme fatigue. However, within only 28 hours of hospital admission, the patient's condition deteriorated into a dual-organ crisis characterized by severe respiratory distress and acute renal failure. Autopsy and subsequent molecular analysis revealed that the virus had not only caused the expected interstitial nephritis in the kidneys, but had also triggered a massive inflammatory response in the lungs, leading to pulmonary edema and shock. This specific case, occurring in a region that accounts for roughly 13% of Russia's total hantavirus cases, has prompted health officials to demand a higher state of awareness for atypical presentations of the disease. Surveillance data from 2000 to 2022 shows that while Russia has historically managed over 164,000 cases of hemorrhagic fever with renal syndrome, the emergence of a strain that can destroy both lungs and kidneys simultaneously represents a significant shift in the biological threat landscape. +1, while the Russian Puumala mutation is being analyzed for its environmental origins in watershed forests, a separate and immediate international response was triggered in May 2026 by an outbreak of the Andes hantavirus aboard the cruise vessel MV Hondius in the South Atlantic. This incident has provided a vital point of comparison for global health agencies like the World Health Organization and the European Center for Disease Prevention and Control. On the MV Hondius, which carried 147 passengers and crew from 23 different nationalities, laboratory testing confirmed that the Andes strain, a species known for its rare ability for person-to-person transmission, had infected multiple individuals. As of May 6, 2026, that cluster resulted in 3 deaths and required the emergency medical evacuation of a critically ill British national to a specialized intensive care unit in Johannesburg, South Africa. Although the Andes virus and the Puumala mutation are distinct species, the simultaneous occurrence of these events has strained international medical logistics and forced a rigorous comparison of viral behavior. +2, the ECDC and the Netherlands National Institute for Public Health and the Environment, or RIVM, have been instrumental in tracking the movement of potentially exposed individuals from these clusters. In the Netherlands, health authorities monitored 3 people who developed symptoms after close contact with an infected individual during air travel, though subsequent tests returned negative. This level of granular surveillance highlights the current sensitivity of public health networks to hantavirus developments. The data currently suggests that while the Puumala mutation in Europe is driven by genetic shifts in the rodent reservoir, the Andes outbreak remains tied to localized environmental exposure in Argentina, followed by human-to-human transmission risks. The contrast between these 2 pathogens is a key focus for lab technicians, One is an evolving endemic threat in the forests of Eurasia, while the other is a known high-fatality pathogen being tracked across international travel routes. This bifurcated threat has led to a push for metagenomic testing in hospitals across the European Union and Russia to ensure that no new reassortant strain goes undetected. Public health guidance regarding hantavirus is being urgently updated to reflect the increased virulence seen in recent mutations. Because the Puumala virus is primarily transmitted through the inhalation of aerosolized dust contaminated by the excreta of the bank vole, prevention efforts must focus on breaking the chain of environmental exposure. Practical takeaways for individuals in endemic areas, which now include much of Central and Eastern Europe, center on a wet cleaning protocol. Health officials warn that under no circumstances should dry sweeping or vacuuming be used in areas where rodents have been present, such as seasonal cabins, sheds, or barns. These actions launch viral particles into the air, where they can remain suspended and infectious for several days. Instead, surfaces should be thoroughly saturated with a 10% bleach solution or a professional-grade disinfectant before any cleaning begins. This dampens the dust and inactivates the virus's lipid envelope, rendering it harmless before it can be disturbed. For those in high-risk occupations like forestry or construction, the use of N95 or FFP3 respirators and non-porous gloves is no longer a suggestion but a critical safety requirement to prevent the rapid-onset respiratory failure documented in the latest clinical reports. Surveillance strategies are also evolving toward a One Health model, which integrates the monitoring of animal reservoirs with human clinical data. Epidemiologists are now calling for systematic genomic sequencing of hantaviruses found in wild rodent populations to identify hotspots of viral reassortment before human cases emerge. The identification of the Moscow reassortant, which targets the lungs and kidneys simultaneously, highlights a significant gap in current diagnostic speed. To bridge this, public health labs are advocating for the inclusion of hantavirus panels in routine screenings for acute respiratory distress syndrome, especially in patients who report recent rural or agricultural exposure. By identifying these mutations early, hospitals can implement supportive therapies, such as extracorporeal membrane oxygenation or early dialysis, which are vital when facing a pathogen that compromises two major organ systems at once. The medical community is also grappling with significant limits to current knowledge, specifically regarding whether this dual-targeting mutation will become the dominant strain or if it remains a sporadic event triggered by specific environmental pressures. The ethical dimension of this reporting involves a transparent assessment of risk without inducing public alarm. Health authorities must balance the need for heightened clinical vigilance with the fact that, for the majority of the population, hantavirus remains a rare infection. However, the documented death of a patient within 28 hours of hospitalization serves as a stark reminder of the virus's potential. There is an ethical imperative for international cooperation in sharing viral sequences, as seen in the collaborative efforts between Russian, European, and South African labs during recent clusters. This transparency is essential for the development of a universal hantavirus vaccine, a goal that has remained elusive due to the high genetic diversity of the virus families. We must also consider the role of human-driven environmental changes, such as land development and climate shifts, which can alter rodent behavior and increase the frequency of human-animal contact. As we move forward, the focus remains on personal protection, environmental awareness, and a robust scientific response to a pathogen that is clearly no longer following the traditional rules of virology. For further analysis on emerging public health threats and global medical reporting, please subscribe to our channel.